Hi all, this is a video where I talk about RuBisCo, an enzyme in the Calvin cycle. It is the second stage of photosynthesis, and it is responsible for carbon fixation, taking carbon dioxide from the air and incorporating it into an organic molecule. We are talking about this molecule, or this enzyme, and carbon fixation because the first step is to deprotonate at the alpha position. We are going to form an enol. But before we get to that, let me jot down a couple of notes about the Calvin cycle and this particular enzyme. Like I said, RuBisCo is an enzyme in the Calvin cycle. The Calvin cycle is considered the second stage of photosynthesis, and there are actually three phases in the Calvin cycle, and the first one is carbon fixation. I won't talk about the other two phases. The Calvin cycle is also considered the light-independent reactions of the plant. RuBisCo, the enzyme itself, is short for ribulose 1,5-bisphosphate carboxylase oxygenase. It is an enzyme that does a whole bunch of very interesting reactions in this one complex. The reaction that we're going to talk about is the first reaction, and we start out with a organic molecule that has 5 carbons. So this is our C5 piece. We're adding a C1 piece, which is the carbon dioxide from the air, and incorporating it into an organic molecule. It's 2 organic molecules, and they're both C3, and they're identical. They are identical. One of them continues on in the Calvin cycle to build up to the C5 piece one more time to start the process all over again. The other half, or the other phosphoglycerate, goes to a different pathway. So this is the reaction where we are going to go through an enolate. Let me scratch that. We're going to go through an enol. We're also going to go through, you might have heard this before in your lecture, the enediol. And somehow we're going to have to get this carbon dioxide to be attacked and incorporated into that molecule before it splits into 2 pieces. Okay, I started on a new sheet of paper because these are pretty big molecules and it is a lengthy mechanism. Now I know I have 2 alpha carbons, but we are going to deprotonate at the alpha carbon on the left, the one in the middle. We're going through a general acid catalysis, meaning that we don't form separately the protonated carbonyl. So this happens at the same time, concertedly, at the rate determined step. And when we do remove this alpha proton and make the double bond here and protonate this oxygen, we do get this enediol intermediate. Okay, let me draw now the next step. Okay, this might actually be shorter than I expected, but in the second step, that's when we incorporate the carbon dioxide. Now, I am remaking a carbonyl group, but I had to choose between this OH or this OH. Choose the one in the middle. If you recall from our picture, the carbon dioxide incorporated into the right side of the molecule, onto this carbon right here. So the idea is to use this double bond, form the double bond here, and then use this pi bond to grab the carbon dioxide. So we did get the CO2 onto this carbon on the right, and we have now this protonated carbonyl group. Next, we do have to break a bond. We now have a 6 carbon piece, and we're going to break it up into 2 3 carbon pieces, and the bond that we're breaking is this one right here. We're going to get that process started by using water. That's an activated carbonyl, so we're going to use water to come in and attach right there. So now we're getting the beginnings of our carbonyl group for the left side of the molecule. Okay, we have quite a bit going on with this intermediate. We did form a tetrahedral intermediate on this sp2 carbon. Now it's sp3. Let me write that down. And probably we're going to deprotonate this group up here to make it the neutral OH. Okay, are we getting anywhere close to a point where we could cleave this bond? Alright, we have this intermediate, and what I would do next is, let's go ahead and break this bond. We'll take the lone pair, make the carbonyl again, and what are we going to do with these two electrons? We're going to put them, let me think about this. Okay, right now we're going to put it onto the carbon. Okay, we have our two pieces. They both have 3 carbons. Just double checking. We have this anion here. Why don't we use that to deprotonate this, and we'll definitely get the carboxylic acid on the left. And let's see what we get on the right. Let's grab this proton. Do I want to grab that? Yeah, I want to grab that proton. And now I think we have 2 identical molecules. I didn't draw any stereochemistry on this carbon here. If we take a look at our final 2 products, they are identical. Okay, so since this is drawn flipped, that becomes dash. I think we're going to do the same thing here. We're going to make this dash. And let me try to attempt to draw the products. Okay, I'm not going to worry too much about the protonation states. Under cellular conditions, that carboxyl group will be deprotonated. I did draw it in such a way that these are identical, so I made this OH the dash. I'm not exactly sure where the selectivity is in the enzyme, what step. Most likely it's going to be here because this one is planar. And then depending on how this hydrogen is grabbed, to push the OH to the back or to the front. That's something that the enzyme can do. We do have our C3 and C3. And let me zoom out and show you the full mechanism. I looked at several sources for the mechanism for RuBisCo, the first reaction. They vary quite a bit. I'm going to go with this. This one has 1, 2, 3, 4, 5, 6 steps. I do hit the key intermediates, which is enediol via a general acid catalysis. And that enediol is the one that, the intermediate that incorporates the carbon dioxide. We do form a tetrahedral intermediate, in this case after the 3rd step. And then we do a couple of proton transfers, and at some point we're going to remake the carbonyl to kick out or to break this bond over here. For my class, I'm not too concerned about the details. I am concerned that my students be able to get to the key intermediates, in particular the enediol and the tetrahedral intermediate, and also do this first deprotonation in this fashion. Alright, not too bad. Just keep in mind what the purpose of RuBisCo, what its job is to do carbon fixation, to incorporate carbon dioxide into an organic molecule. And it starts out by first deprotonating at the alpha carbon.